Alrighty. So let's get started. So second to last class. So that's pretty exciting. It's pretty exciting indeed. Now today we were covering photosynthesis and respiration, and in terms of turnables in, there's nothing really due this week, which is it's always good. And there is an in-class activity doing some genetic counseling. Now, the following week, we're going to cover energetics and metabolism, and then we have our final in-class activity, how much photosynthesis and respiration occur in the global ocean, which is a fun little activity that I wrote about using some what we call back-of-the-envelope calculations to estimate photosynthesis rates. So I think it's pretty fun. Now, I was looking at the schedule, and I had a thought, and I wanted to run it by you guys. So we only have one class next week. Now we have 2 hours today. So my question to you guys, would you rather we do class this week and then class next week? Or do you want me to pack in respiration, photosynthesis, and energetics into one class, and then you do the in-class activities on your own? So I'm gonna give you guys a poll. So it's up to you guys. We can have a longer, like we'll pack in 2 lectures in one day, and then you just do the class activities, genetic counseling, and photosynthesis on your own and submit them as a discussion board, and we won't meet next Wednesday. So it's up to you guys, whatever you want to do, but I figured I'd make the offer since, you guys have other stuff, right? You have your third exam, you have your BioWiki final draft, and you have one homework, and you have extra credit left. So I figured you guys might want a little bit of extra time to do all those things that are due. But again, it is up to you guys. You guys know you gotta when you're here, you actually gotta pay attention. You can't just step away from your computer, right? All right, so the yeses have it by a large majority. So that's cool. It's 10-2 since some people didn't bother to respond. All right, so we'll talk about metabolism and energetics and photosynthesis respiration today, and your in-class activities. So for this week, it's genetic counseling, and then the following week, it's respiration photosynthesis. You could do those on your own time, and there are discussion boards. At least there should be for them. I'm pretty sure I set them up. Yeah, so how much photosynthesis, and then genetic counseling. So both of those are there for you to submit your stuff to. So you still got to do them, even though we're not going to meet. And like I said, I like the photosynthesis one. It's this really cool estimation of photosynthesis, and it's actually kind of, I thought it was pretty cool, but I wrote it, so I'm kind of being biased here. So all right, so we won't meet for class next Wednesday. Today will be your day. You should turn them in by next class. So by the time next class should have ended, i.e. at 4 o'clock, you should put them in there. They're only designed to be about 20 minutes long anyway. So all right. So anyway, so fun stuff today. I really like this series of classes that we're going to cover today. I really do. It's really fun. And I'm a little bit biased because when I was a graduate student, I studied a bacterial process called denitrification, which is a type of metabolism, but it's only performed by bacteria and a very small set of fungi. So I'm really into metabolism, I'm really into energetics, and I'm really into respiration. So I love these couple classes because it ties directly into what I used to do for research. I've probably said I'd love classes a lot throughout this semester, and that's just because I like biology a lot. So. Anyways, so we'll start off with energetics and metabolism. So when we're thinking about this, all life pretty much exists to do a couple sort of different things, right? It's to pass on genetic information, and we discussed that last week with meiosis and sexual inheritance and all that gobbledygook. But the other thing life it really does is just kind of processes energy, and we kind of put it to the back of our minds, right? We think about energy in terms of like I turned on a light bulb or I drove my car to the store, right? That's energy. But we also think of energy in terms of life, right? You eat food for energy, right? So energy and life are directly tied. So we're going to go over all this fun stuff. But before we get into energy, we need to think about one of the sort of, not the biggest, but one of the more important areas of chemistry, which we call the laws of thermodynamics. Now, thermodynamics basically, it sounds, I don't know, I always thought it sounded kind of like an intimidating kind of word, right? But if you break it down, dynamics is how things change, and thermo is just refers to energy. So when we look at thermodynamics, it's simply the flow of energy. So that's one. And everything in the universe, whether it's a star or whether it's you and me or your cat, is governed by the law of thermodynamics. Let me see. Mute all. You guys gotta remember to turn your mics off if you're not speaking. So that's what we're thinking about. Now we define energy, and if you've taken chemistry before, this will sound just repetitive, but chemistry tells us that energy is simply the capacity to do work. And what does work mean? Work just simply refers to moving anything, right? The movement of anything. And this can be movement of you across the room. This can be the movement of a molecule inside you. But all work describes is the capacity to have energy, i.e. motion. And when we describe energy, we describe it in two distinct states, and they're both related to one another perfectly. Kinetic energy describes the energy of motion, and this is what we think about as work. If you're doing work, you have kinetic energy, but kinetic energy simply refers to the movement of objects. So if you are running on a treadmill, you have kinetic energy. Now, the opposite of kinetic energy is what we call potential energy, which is essentially stored energy. And it's the capacity to do work or the capacity to gain kinetic energy. So when I hold pen in my hand, I don't know if you guys pay attention to my face cam when I do this, but when I hold a pen in my hand, the pen's not physically moving, but it has the capacity to fall out of my hand and gravity will pull it down towards my desk. So my pen has the potential energy to move, right? And so it's the capacity to do work. Now, when we think about potential energy in a biological context, we're typically speaking about the energy stored in food. So when you eat a sweet, awesome Chipotle burrito bowl, I ate that for dinner last night. It was pretty good, right? There's potential energy stored in that Chipotle burrito bowl that as you digest it and metabolize it, you extract that energy. But again, it's all related to the fact that there's the capacity in food or any substance to do work or to have kinetic energy. Now, in these two categories, there's lots of forms that energy can take. It could be mechanical, it could be heat, it could be sound, it could be an electric current, light, or even radioactivity. So there's lots of modes by which energy can take, or I'm sorry, shapes by which energy can take form. So it's not just thinking about, Chipotle burrito bowls or anything like that, right? So, but when we think about biology, heat is one of the most convenient ways to measure energy and one of the most common ways energy is produced. So if you think about when you are just doing your average everyday thing, right, your body exists at a consistent 98.6 plus or minus a half a degree. How is it that warm all the time? Well, you're producing heat energy, right? So when we measure energy, we most commonly measure energy as a calorie, which is defined as the heat required to raise 1 gram of water by 1 degree Celsius. And again, if you've taken chemistry, you've heard this term before, especially if you've taken it with me. Now, when we look at calorie, just as a note, when we look at calories on food labels, you'll notice that those have a capital C. Calories on food labels are actually what we call kilocalories, which are 1,000 calories. So 1 calorie when you eat it, i.e. on like, you go out and buy a Snickers bar and it says there's 300 calories. What that refers to is 300 kilocalories, which would be the capacity to heat, in this case, 300 kilograms of water by 1 gram. Sorry, 1 degree Celsius by 1 gram. Idiot. So anyways, but again, just to reiterate what potential and kinetic energy are, potential energy is the capacity for this kind of evil looking girl to go down the slide, and then kinetic energy is her physically going down the slide. I don't know what it is drawing kids' faces, but man, they always look creepy in textbooks. I don't know what it is. It's like an illustrator thing. I don't know, but that's just me. It's the end of the semester. It's been a busy semester for all of us. So now, when we're thinking about biology, energy flows in the biological sense. It starts at the sun, and that energy from the sun basically flows from the sun to organisms, and those organisms capture that energy, and those organisms are photosynthetic, so plants and algae and things like that, and they convert that energy from the sun into potential energy in the form of chemical bonds. Now, these chemical bonds are in the form of sugars. So you're thinking about glucose, right? Things that we've already discussed, right? So we're taking energy from the sun, we're capturing it in organisms, and those organisms are converting that energy in the sun into sugar, which has potential energy. Because again, what is really potential energy in a biological context? Well, you consume food, you break it down, you get energy, and you do all your things with that energy. So that's the flow of energy. Sun to photosynthetic organism to everything else. Now, that's the, well, let me take one step back. That's how the vast majority of energy moves. Now, there are other forms of energy that biology can harness, but we're not going to talk about them, just as a note. But like, there's weird stuff that goes on at the bottom of the ocean and deep underground where you get energy from other molecules. But by and large, when we're thinking about the flow of energy, it's sun to plants to everything else. So that's the flow. And this allows us to talk about a very specific type of chemistry, and chemistry that, again, if you've taken chemistry, will sound very familiar to you, but it's what we call redox reactions. Now, redox reactions simply refer to the oxidation and the reduction of two distinct molecules. So when a molecule is oxidized, it loses an electron. When a molecule is reduced, it gains an electron. And the interesting thing about reduction is that things that are reduced, i.e. that have gained an electron, have much higher energy levels. So oxidation and reduction, when you pair them together, are what we called redox reactions. And by and large, I can't think of an exception to this rule. That's why I wrote it like this, but they are always paired. So if something is going to be reduced, something else has to be oxidized. If something is going to be oxidized, something else has to be reduced. So they are always paired. So what does this conceptually look like? We start off with A and B. A transfers an electron to B, thus by A becoming oxidized and B becoming reduced. And A, because it lost an electron, has lost energy relative to A on this side. And B, because it has gained electron from A, has higher energy than it did on this side. So by transferring an electron, we transfer energy. Now, it's a little bit different than this analogy I'm going to use, but when you charge your cell phone, for instance, you plug it into the wall. Well, what comes out of the wall socket? Electrons. Electrons are transferred from your wall socket into your phone, thus putting your phone at a higher level, right? The battery takes up that energy. So that's the sort of the flow we're thinking about, right? When something becomes reduced, it gains energy. When something becomes oxidized, it loses energy. So this leads us to the first law of thermodynamics. Now, we have a law here. And remember, laws are the highest level of scientific inquiry. A law is consistent across the entire universe, across all throughout history. Laws are they can never be violated. They are always the way they are. So the first law of thermodynamics is the same here on Earth as it is on Mars, as it is in the sun, as it is in the Andromeda Galaxy. Laws are consistent through space and time. So our first law of thermodynamics easily enough states that energy physically cannot be created, nor can it be destroyed. Energy itself can physically change from one form to another. However, the total amount of energy in the universe has to remain constant. And the final thing is that during each conversion, some energy is physically lost as heat. So let's put this in a biological context. Energy that a plant takes in was not simply created, it existed before, but the plant takes the energy in the form of light from the sun, changes its form into sugar, that's photosynthesis, which we'll talk about in a little bit. So we're changing it from light energy to chemical bond energy, but that amount of energy from the sun to the molecule has to remain constant, right? There's no net increase in energy in the universe. However, as we convert that energy from the sun to sugar in the plant, some of that energy in the sun was lost as heat. So that's what we mean in terms of putting this in a biological context. Right? Energy can't be created or destroyed. And there's another law out there that says matter cannot be created or destroyed. It's very similar. You can't create or destroy things in the universe, not truly. So kind of cool. Now, second law of thermodynamics, which is probably the hardest one to understand, but we have entropy, which is simply defined as disorder. And when you say disordered, as in like, if I have a box of Skittles and I throw those Skittles on the floor, that's disorder, that's entropy. But entropy in the universe itself is always increasing. This one, like I said, this one's a little bit hard to grasp, but just conceptualize what I'm thinking about here. Now, energy transformations themselves do proceed spontaneously to convert matter from a more ordered / less stable form to a less ordered and more stable form. So let me give you an example of what entropy looks like in a context we all know. We have some order here, right? So we have, when we're thinking about this, right, we're going from more ordered, less stable to less ordered, more stable. Very, very ordered. Ice cubes are very, very ordered, but they're not very stable, right? They melt. So as the ice cubes start to melt, entropy increases. The disorder, right, is increasing, but this is a much more stable system when the ice is physically melted. Why is it a more stable system? There's no exchange of heat between the ice cubes and the water around them anymore. So by melting ice, we go from something that's less structured, but it becomes more stable in an energy standpoint. So again, kind of a weird concept, but definitely something we need to think about, because biology does this all the time, right? We take energy from the sun, we convert it into sugar, and there is a change in entropy between those two processes. Or when I eat a Chipotle breedable, I convert the energy in my food to energy that I can use, and there's a change in entropy between those two. All right. Now, we're going to define a new term, which we call G, or it's also called Gibbs free energy, but we're going to define it as G, or the energy available to do work. Now, G itself is equal to H minus T times S. You guys thought you were going to get away from math this semester in this class, didn't you? Surprise. But we define G, H, which is enthalpy, which is the energy in a chemical's bonds. I'm sorry, molecules bonds. T, which is the absolute temperature, and S, which is the entropy, which is the amount of unavailable energy. So that's G. Now, more importantly, though, we can calculate what we call delta G, which is the change in free energy. And I'll show you what this looks like in just a second, but let's conceptualize it first. Now, G itself is, again, this energy available to do work. When we calculate delta G, what we're really looking at is the temperature is going to remain constant, as is the entropy. But what's really going to change is H, right? So when we're looking at the change in free energy available, when we look at a positive delta G, the products, i.e. what's on the right side of a chemical reaction, has more free energy than the reactants, which is on the left side of the equation. H itself is physically higher, or entropy, S, is physically lower. Delta G is not spontaneous, which requires, the reaction itself is not spontaneous, which requires an input of energy, which makes this reaction what we call endergonic, i.e. it consumes energy. Now that's in contrast to a negative delta G, which we define as the products having less free energy than the reactants. H itself is lower, or S has to be higher, and it's spontaneous. It might not be instantaneous, but it's certainly spontaneous, and it's exergonic, i.e. it releases energy. So if we're going in a reaction from left to right, a negative delta G releases energy, a positive delta G consumes energy. And that's the way we're thinking about this. So let's physically just visualize what this looks like first. So we have our friend here, G, all right, our free energy for both A and B here, where the reactants, i.e. the left side of the equation, is, where delta G is greater than 0. So we see that the energy in the reactants is less than the energy in the products, which means to push the reactants, i.e. the left side of the equation, to the right side of the equation, energy physically must be supplied. Now that's in contrast to a negative delta G, where there's much more energy on the reactant side, i.e. the left side of the equation, than there is on the products, i.e. the right side of the equation. And when a delta G is in fact negative, energy is released. So keep that in the back of your mind. We'll come back to it in a little bit. Now, next topic, what we're going to discuss, what we call activation energy. Now, activation energy is simply the extra energy required to destabilize existing bonds and start a chemical reaction, i.e. what energy is needed to push a reaction from products to reactants. That's really what we're getting at. Now, the rate of an action that's exergonic, and when we're thinking of exergonic, it's releasing energy, depends on the activation energy required. Now, the larger the activation energy, the slower a reaction ultimately proceeds. And activation energy can be altered, or the rate of reaction can be increased by either increasing the energy, i.e. heating, or lowering the overall activation energy. So let's look at what this looks like. Now, we're looking at our friend free energy here, and we have the course of the reaction. We have the reactants, which we start, i.e. the left side of the equation, and then we have our products, right, i.e. the right side of the equation. Now, we can look at conceptually, delta G just refers to the change in energy between the products and the reactants. Now, we can look at two distinct things. We can look at a uncatalyzed versus a catalyzed delta G, right? Catalyzed or uncatalyzed activation energy. And what do I mean by that? Well, let's first just talk about what the activation energy is really getting at. Now, higher the activation energy, the more energy is required to physically push a reaction forward, i.e. going from products to reactants. So in this case, we have catalyzed versus uncatalyzed. So uncatalyzed is in blue, catalyzed is in red. You notice that when you have a catalyst present, it lowers the activation energy, i.e. there's less energy required to push a reaction forward. Now, what is a catalyst? In a biological context, we think about a catalyst being an enzyme, right? Enzymes are just specific classes of proteins that help catalyze chemical reactions. How do they help catalyze chemical reactions? Well, they lower the activation energy. So if you ever wondered what an enzyme physically does in a chemical reaction, really all it does is just helps lower the activation energy. Oftentimes it just puts two things close together so that they are more likely to collide, thus react. It's really all most enzymes do. So that's our activation energy. But again, we can lower activation energy by using a catalyst. And the broad definition for a catalyst is substances that influence chemical bonds in a way that lowers activation energy. Now, biological speaking, it's enzymes. There are other types of catalysts out there. You know, there's metals and other compounds that help, non-biological reactions proceed. But biology, we're thinking about enzymes. Now, catalysts, they sound great, right? They can lower activation energy, they can help push reactions forward, but as with everything in the universe, they cannot physically violate the laws of thermodynamics. So they can't make an endergonic reaction, i.e. a reaction that requires energy to be spontaneous, and they can't alter the proportion of reactant turned into product. So they do a lot, but they can't violate um, these rules. And so again, showing you what a catalyst looks like. Activation energy, i.e. the amount of energy needed to push a reaction forward. Catalyzed, much less activation energy than uncatalyzed. So you can think about this as a reaction inside your body. To break down, say, glucose, you need an enzyme to do it. Well, what that enzyme is doing is lowering the energy required to break down that molecule inside your body, i.e. glucose. Okay, so that's like the part that you probably hated. That's the chemistry E stuff. So let's talk about what biological energy physically looks like. And it's a really nice acronym, ATP, adenosine triphosphate. The adenosine part should jump right at you. Where do we know adenosine from? Well, it comes from our friend adenine. And the chemical way by which cells carry energy is via ATP, adenine. So the DNA base that you know and love, which is bound to that 5-carbon sugar, is bound to 3 phosphate groups. A at adenine, tri 1, 2, 3 phosphates, adenine triphosphate or ATP. Now, just as a note, a phosphate group is simply PO3 or PO4, just as a note. So PO3 you see here, phosphate. That's why it's triphosphate. So pretty cool, right? Very simple molecule, but very powerful way to carry energy because ATP carries energy, but it carries it carries two very high energy bonds between the first ATP and the second ATP, and between the second ATP and the third ATP. So first, second, third ATP, these two bonds, as noted in this figure, carry a lot of energy. And when you break those bonds, you release energy, and you release the energy to do something inside your cell. Now, you'll also recognize ATP from our cellular signaling class, right? When we talked about phosphorylating something, where did we get that phosphate group from? We got it from our friend ATP. So ATP plays a lot of roles. Right? It's not just involved with signaling pathways, it's involved with the direct transmission of energy. Now, when we break ATP down, so when we lose a phosphate group, we become ADP, or adenosine diphosphate, because we have not 3 phosphates anymore, but 2 phosphate groups. And when we lose 2 phosphate groups, i.e. we just have 1 phosphate group left, we become adenosine monophosphate. And so as we go from mono to di to triphosphate, we increase the amount of energy. So low energy with mono, medium energy with di, and a lot of energy with tri. But once you are at AMP, there's no energy to be gained by getting rid of the third phosphate. All the energy is really stored in this bond and this bond itself. So that's our friend ATP. Know him and love him. He will serve you well going forward. Just as you know, when you take cell biology, you would talk about ATP a lot, just because of how important it is. So know ATP, love ATP, and just remember it. Now, ATP, because it's so important to the biology of a cell, it has its own little cycle inside of your cells. And it's not just your cells, it's also bacteria and fungus and everything else. Because ATP is the energy currency not just for you, but it's the energy currency for all of life. I don't think I've mentioned that before, but it is the energy currency for all of life. Now, the hydrolysis of ATP drives endergonic reaction. What do I mean by hydrolysis? What I mean is simply by cleaving off, or i.e. breaking off one of these phosphate groups. That's what we mean by hydrolysis. Breaking off a phosphate group is hydrolysis of ATP. So what ends up happening is when you hydrolyze ATP, i.e. you take a phosphate group off ATP and you stick it on something else, you transfer energy. Now, what does that do conceptually to the chemistry? it results in a net negative G, so free energy, which makes the reaction exergonic and spontaneous, i.e. likely to happen. So our ATP is related directly to our G, right? When you have ATP present and it's hydrolyzed, i.e. it loses a phosphate group, it transfers energy, thus lowering the G in your system, thus making the chemical reaction likely to happen. Now, ATP is great. It's really great, but ATP is short-term storage of energy. The phosphate bonds themselves are pretty unstable. So if your cell is going to store something for long-term, it stores it as fat and carbohydrates. In the case of human cells, we store most of our energy, at first at least, as glycogen, just as a note. And as a whole, when you actually do the math behind this, cells only store what we call a few seconds of ATP, which is basically, it's a weird way to think about it, but it's like, if you think about it like a battery, right? Think about you have a battery, but it only powers your cell phone for like 3 seconds. That's how cells store ATP. They basically have enough ATP on hand to power themselves for a few seconds, which again, which I think is a weird way to think about it, but when you do the math behind it, it's actually what you end up getting. So it's actually kind of cool. So let's look at the ATP cycle. If we have ATP, we have ATP in water. If we cleave off one of those phosphate groups, i.e. we hydrolyze it, it provides energy for endergonic cellular processes, i.e. lowering the delta G values. Now, to make ATP, all we do is simply take ADP and we tack on a phosphate group. We technically called phosphorylating it, and that requires energy to do. So to add a phosphate group to ADP physically requires energy. Who would have thought? So that's our ATP cycle. Very straightforward. You either have ATP or you don't. And to get ATP, you have to stick on a phosphate group. And to do that, you have to use energy. So as I've mentioned, when we think about catalysts, biologically speaking, it's enzymes, right? Our special class of proteins that are involved in chemical reactions. Now, by and large, enzymes, as I mentioned, are proteins. There are some enzymes which we call ribozymes. We're not going to talk about them, but they are called ribozymes, and they're just made out of RNA. RNA is kind of a cool molecule. As you'll learn in biology too, you'll learn that early life was all RNA based, because RNA is a catalytic molecule. So it doesn't just store energy. Store information. It has the capacity to do chemistry as well. So RNA, as what we call a ribozyme, can actually function as an enzyme. So just wanted to make mention of that. It's a pretty cool thing that RNA does. RNA is a wild molecule, one of the more wild things out there in biology. At least in terms of the basic things. Now, the shape of an enzyme physically stabilizes a temporary association between two substances. So if we're reacting glucose with something, all an enzyme does is really just brings them together in a physical space. Now, once they're physically together, they can react chemically. Now, when we're thinking about what an enzyme does, it doesn't change shape. It's not consumed. It doesn't get destroyed or anything like that. It's just there to help chemistry proceed, right? It's a catalyst, thus it is lowering the activation energy. Goddamn fly. So for example, I'll give you an example of an enzyme called carbonic anhydrase. Carbonic anhydrase is involved with, as it suggests, is with CO2, carbonic part. Now, enzymes are so good that without carbonic anhydrase, only 200 molecules of carbonic acid per hour are made without the enzyme. With the enzyme present, you can form upwards of 600,000 molecules per second with this enzyme. So 200 molecules per hour, or with the enzyme present, 600,000 molecules per second, which is wild, right? You're going up 5 orders of magnitude there. That's crazy, right? But but enzymes are fantastic, right? They make chemistry happen. Now, we can physically look at an enzyme. This is just an arbitrary enzyme. Sorry, there's a fly flying around me. It's to drive me crazy. Anyways, we can look at an enzyme. It's just some arbitrary enzyme, right? It's likely a protein. It has what we call the active site. And the active site is where our substrates, i.e. the chemical reactions we're trying to catalyze, are going to bind. Now, all an enzyme does is just have to have this active site. And the substrate will fit perfectly into this active site, thus causing the enzyme to change its shape slightly, forming what we call the enzyme substrate complex. Now, in your lab with amylase, amylase was the enzyme, the substrate was starch. Starch fits directly into the active site. Amylase changes conformation or shape slightly. That change in shape causes the enzyme to break down to break down starch into sugar monomers, right? So that's the sort of the flow. Now, as I mentioned, the active site is really just kind of like a pocket. You can also think of it as like a cleft, but all it's really used is for substrate binding. And it's a very precise fit. So if you are an enzyme that only catalyzes the breaking down of starch, well, nothing else is really going to fit in there, only really starch, which is, I think it's a good thing, right? You don't want some enzyme that's used to break down starch to start breaking down your proteins or something like that, right? So enzymes are pretty specific in what they do. But from a physics standpoint, what the enzymes really do is they basically just kind of apply a stress to distort a bond or to basically lower the activation energy. So they bring things together or they kind of distort things to make things happen. But let's look at this. I think this is a great example because this enzyme is what we call sucrase. Now you have sucrase inside you. It's a pretty common enzyme. Now sucrose is just a monomer, a disaccharide, which is comprised of a subunit of glucose bound to a subunit of fructose. So this is a disaccharide because there's two sugars. Now, sucrase has an active site and it has this general shape, kind of looks like a W. But you'll notice that the active site is perfectly attuned to the shape of our glucose and our fructose, i.e. sucrose, bound together. Sucrose is table sugar, right? So if you put it in your coffee or tea, you put sucrose and you use this enzyme, just as a note. Now, what sucrase does is once these are bound, it consumes some water. So it does what we call a hydrolysis reaction. A dehydration reaction where it consumes water. And it cleaves glucose and fructose physically apart. So what it does in this case is it places essentially amount of stress on this glucose-fructose bond here in white to break it in half. Then once it's broken it in half, it releases the products, i.e. glucose and fructose, into the cell, and then it frees up its active site to repeat this process over and over again. So you notice we don't break the enzyme, it's reusable, right? We don't do anything to it. And this reaction can essentially happen for an extended period of time. Now, it can't happen indefinitely because enzymes will eventually start to break down, but they last for a decent amount of time. So this is our cycle of enzymes, pretty straightforward, which is pretty good. Now, enzymes themselves can be either floating around in the cytoplasm or be attached to cellular membranes or organelles. And they typically form what we call multi-enzyme complexes. So we're thinking about, in terms of protein structures, we're thinking about that quaternary structure, so the highest level of protein structure. And the subunits themselves physically work together to form a molecular machine. And so basically, if you have one process, i.e. the breakdown of a sugar, and you need multiple enzymes to do it, they typically form these multi-enzyme complex. Or if it has to go from enzyme A to B to C to D, they simply associate with one another and thus can pass things to one another to control the reaction as a unit and speed up the reaction by physically associating these enzymes with one another. So that's typically what we see. Now, as I mentioned, there are ribozymes. They do some stuff, but we're not going to talk about them, but I just figured I'd leave this information there for you as well. Now, the rate of enzyme catalyzed reactions physically depends on the concentrations of the substance of an enzyme And it also relies on the physical conditions that affects the enzyme's physical shape. So temperature and pH do this. So the environment affects enzyme activity, and the concentration of the substrate also affects enzyme activity. The other thing that also happens, as you guys should remember from your amylase lab, as you increase the concentration of an enzyme, it also speeds up the enzyme. So the more enzyme you have, the more substrates you have, the faster reaction will go. However, hotter temperatures, as you know, increase enzyme activity. Lower pHs, higher pHs also change enzymatic activity as well. So we can visualize this, and you guys sort of got to see this in your amylase lab. But you have the rate of reaction, and we have temperature and pH. Now you'll notice that we have two distinct enzymes, one that likes to operate at low pH, high pH, medium temperature, and then very hot temperatures. And you'll notice they follow like these bell curves, right? Nice curve. And you'll notice that they've all been labeled as optimum temperature and optimum temperature, right? Or optimum pH. Right, so enzymes have a range of pHs and temperatures that they can operate on, but they have what we call an optimum, which means they operate the best at that temperature. They catalyze reactions faster at that temperature. So you can see, like, for instance, the optimum temperature for a human enzyme is about 37 to 40 degrees. The optimum temperature for something that lives in a hot spring is closer to 70 degrees. So it just depends on the organisms. Now, the another part, again, which we covered in the amylase lab, but we have what we call inhibitors, and these are substances that physically bind to an enzyme and decrease its activity. So we have competitive inhibitors, which competes with a substrate for activity. So this would have been like in your lab, it would have been like one of the amylase blockers, or it would have been uh, um, like SDS, for instance. And then you have what's called non-competitive inhibitors, which physically bind to an enzyme at a site other than the active site, which causes a change in the shape, which makes the enzyme unable to bind to the substrate. So let's look at this. So competitive inhibitors, where you have an inhibitor directly binding to the active site, and then you have the substrate itself physically can't get in there because the inhibitor is there. Now, conversely, if you have a non-competitive inhibition, you can see the active site is freed up on the enzyme, but there's a site which we call the allosteric site that is bound by an inhibitor. Now, because it's bound by the inhibitor, you see it changes the shape of the enzyme from, it started like this, and then it kind of goes to this. Thus, the substrate physically can associate with the enzyme. So fun stuff. Now, allosteric enzymes exists in both active and inactive forms. So active is when the inhibitor is not bound. Inactive is when the inhibitor is physically bound. Now, most non-competitive inhibitors bind to the allosteric site, so it's basically like a chemical on and off switch. We have allosteric inhibitors, which binds to the allosteric site and reduces enzymatic activity. And then we have allosteric activators, which binds to an allosteric site and increases enzymatic activity. So three distinct things underneath are sort of allosteric enzymes. So, and that should be near the end. All right, so that brings us, all that chemistry stuff, that brings us to metabolism. Now, metabolism is simply the total of all chemical reactions carried out by an organism. We define them as two distinct classes, anabolic or anabolism, which are energy dependent, and they're helped used to build molecules or help to build biomass, i.e. like, parts of your cell or muscle or whatever. Or we have catabolic reactions or catabolism, which harvests energy by breaking down molecules. So those are our two distinct things. Catabolism and anabolism. Anabolism consumes energy, catabolism releases energy from stored things. Now, most of our metabolism is oriented in what we call biochemical pathways. And these basically are reactions that occur in a sequence. And so we'll talk about a bunch of biochemical pathways in a moment. But basically the product of one reaction is the substrate for the next reaction. And many of them, as you'll learn, takes place in specific organelles or in specific locations in the cell. But I'll give you just a broad example. We have the initial substrate, it binds to enzyme 1. There's intermediate produced, that intermediate binds to enzyme 2. produces intermediate B, and then so on and so forth, leading to a final end product. So that's what we think about when we think about a biochemical pathway. Now, um, feedback inhibition, which is basically the end product of a pathway, increases in concentration as it's synthesized. So what does that mean? As this biochemical pathway occurs, we're producing more and more of this end product. Now, the more product increases, the probability that it binds to an allosteric site on an enzyme in the pathway, and it causes that physically to change shape so it cannot bind its normal substrates. And this essentially shuts down the pathway so raw materials and energy aren't wasted. So what does that mean? Well, if we have this pathway with enzyme 1, 2, and 3, it takes the initial substrate here, which is in orange. Now, and it produces an end product, which is in green. Now, feedback inhibition says the more green we produce, the more likely it is for that green to bind to that allosteric site on our enzyme, thus causing a change in enzyme 1, thus leading to the substrate being unable to bind. So as we have more and more and more of this end product, well, we really don't need to break down more of this initial substance, right? That's just a waste, right? If you if you already have like a birthday cake worth of cake that you're going to eat by yourself, well, do you really need to keep baking birthday cakes for yourself? Probably not. Thus, you inhibit yourself from baking a birthday cake, right? And that's the same thing here. If we're breaking down something into an end product, well, if we have lots of that end product, we don't need to keep breaking down that thing. We should stop. And the way we stop is by this feedback inhibition. All right. So that is part 1. Fun stuff. It's always fun when we talk about chemistry, I'm sure. But all of that leads us into our friend photosynthesis and respiration. So chock full of things to talk about now, but they are biologically very important things. Now, we can classify organisms based upon their capacity and how they obtain energy. So we define organisms as autotrophs, which are organisms that are physically capable of producing their own organic molecules through either what we call photosynthesis, using the sun, or what we call chemisynthesis, which is a different type of autotrophy, which we won't discuss. But I figured I'd make mention that photosynthesis is there, but there's also chemisynthesis as well. And then we have heterotrophs, which live on organic compounds produced by other organisms. That's us. So the plants we eat are autotrophs, and we are heterotrophs. That being said, all organisms, whether it's a plant or whether it's you, use cellular respiration to extract energy from organic molecules. Now, cellular respiration physically describes the process by which we extract energy from chemical bonds, i.e. sugars, fats, and things like that. Now, as we've mentioned, when something is oxidized, it has lost electrons. When something is reduced, it has gained electrons. And then the final term that we'll see in cellular respiration is what we call dehydrogenation, which is the loss of electrons that is accompanied by protons. So what does that mean? We lose, basically every time we lose an electron, we also lose a proton along with it, just as a note. So as we mentioned, redox reactions, i.e. something gets oxidized and the other thing gets reduced. Electrons themselves are physically carried from one molecule to another. Now, electron carriers are an important part of cellular metabolism or respiration. And one of the more important ones is what we call nicotinamide adenosine dinucleotide. I'm never going to say that again. It just simply refers to as NAD. Now, NAD and its friend NADH, NAD and NADH, is what we call an electron carrier. It's also what we call an enzymatic cofactor, but that's less important. Now, NAD accepts 2 electrons, i.e. it becomes reduced, and it also gains a proton to become NADH. Now, this reaction itself is reversible, so NADH can lose 2 electrons and lose a proton to become NAD again. But when we add 2 electrons, NADH serves as an electron carrier. Because remember, when you reduce something, i.e. you give it electrons, it gains energy. And if you gain energy, you simply can transfer that energy away if you so choose. So that will become important in just a minute. But this is the flow that we've discussed before, oxidation and reduction, what this physically looks like. So I'm not going to go through redox again, but this is, again, what's going on. Now, redox reactions, in a overall biological sense, help cells harvest energy. But these redox reactions aren't just one. We're typically thinking about dozens of chemical reactions that have to occur for you as an individual at the cellular level to extract energy from something you eat. During all this process, because we have a redox reaction, electrons have to be transferred. And we have to have a number of electron acceptors to accept electrons from whatever is being oxidized. So by the end of this, high energy electrons from the initial chemical bonds, i.e. from, say, glucose or sugar, basically lose much of their energy. But ultimately, you transfer that energy to what we call a final electron acceptors. Now, what is a final electron acceptor? Another way to say a final electron acceptor is also the terminal electron acceptor. What is the last thing in your respiratory chain to accept the electron? Because remember, all respiration is just a bunch of redox reactions happening over and over and over again. And so electrons have to be transferred from one molecule to the next. But there has to be an end at some point, and that end point is the terminal or the final electron acceptor. Now, the final electron acceptor varies. Now, in aerobic respiration, i.e. a respiration in the presence of oxygen, the final electron acceptor is our friend oxygen. Now, in anaerobic respiration, where there is no oxygen, the final electron acceptor is an inorganic molecule, but it's not oxygen. So this can be sulfur, it can be nitrogen, it can be iron, it can be manganese, it can be any number of things. And then finally, we have fermentation, which the final electron acceptor is a physical organic molecule, like a sugar or carbon dioxide or something like that. So again, metabolism, bunch of redox reactions happening over and over again, but you have to transfer that electron to something at the end for all this reaction to stop. So these are our terminal or final electron acceptors. So aerobic respiration, which we're going to spend the most of our time talking about, can be simply broken down into this very, very simple chemical equation. Glucose, C6H12O6, reacts with 6 moles of oxygen, 1 mole of glucose, 6 moles of oxygen, to produce 6 moles of carbon dioxide, 6 moles of water, and free energy. So when you eat a candy bar, you break down the sugar in the candy bar with the oxygen that you breathe to produce CO2, which you exhale, water, which is just inside your cells, and energy, which you use to power your cells, i.e. listen and speak and all the fun stuff. Now, the free energy, or the G for this, is negative 686 kilocalories per mole of glucose. Now remember, when G is negative, it's a reaction that likes to happen. So we can get a lot of energy for it. Remember, the smaller G is, the better the reaction. Now, this is when we look at from a chemical standpoint. Now, the free energy can be even higher than it is in this physical cell. Now, this large amount of energy has to be released in fewer smaller steps. So this is like the overall reaction. You have to do this piece by piece because you can't do it all at once. But the whole reaction itself has a negative delta G of 686. And how do we? What are some sort of the steps along the way? And I don't mean actual physical steps. Like, what are some of the steps by which we do this? Now, as I mentioned, metabolism is just a bunch of redox reactions. We're transferring electron from something that's oxidized to something that is reduced, right? So we have to transfer this. This is what we term, generally speaking, as electron transport. Now, we start off with electrons from food, and we transfer them from one substrate to the next, to one molecule to the next, to the next, to the next, to the next, eventually getting us to our final electron acceptor, which is oxygen. Now, along the way, we're going from very high energy to very, very low energy. And along the way, releasing energy to power ATP synthesis. Now, remember, ATP is the energy currency from the cell. So we're taking the high energy electrons from food, passing them from molecule to molecule. Along the way, we're releasing energy to generate ATP. And then we finally cessate the reaction or reaction by transferring it to oxygen and forming water. So, but for this this process to happen, as I've mentioned, we have to have electron carriers. Now, as I mentioned, NAD is an electron carrier, but there are many other types of electron carriers inside of your cells that are used, and they all are oxidized or reduced at some point, but they can be reversibly oxidized or reduced. And they have multiple functions usually. They can carry electrons. Some can carry electrons and protons. But again, they're all involved in this series of transferring electrons. Because remember, transferring electrons, oxidizing and reducing things, that's what metabolism is all about. So I just want to show you what NAD looks like before we sort of go on. Now, this is NAD. This is oxidized NAD plus or NADH. Now you'll notice they have the same structure: adenine, 5-carbon sugar, phosphate, 5-carbon sugar, which is then bound to a nitrogenous ring here. Now the difference between the two of them, as you'll notice, isn't a lot, right? We have N2, NH2, CO, right? Going from oxidized and reduced, there's not a lot difference, right? We tack on a hydrogen, right? And that's pretty much it, right? There's not a whole lot of difference between NAD and NADH, but this one is reduced, this one is oxidized. So this one is carrying an electron, this one is not carrying an electron. So as I've mentioned, ATP is what is used to drive our endergonic reactions forward. As we mentioned, delta G, or the free energy of essentially tacking on of removing a phosphate group from ATP, i.e. generating energy, has a delta G of minus 7.3 kilocalories per mole. So there's a lot of energy in just that final um, um, phosphate group in the ATP. Now, to generate ATP, there are two primary mechanisms. We have substrate level and oxidative phosphorylation. Now, substrate level phosphorylation is where we have a transfer of a phosphate group directly to ADP, and this happens during glycolysis. And then we have oxidative phosphorylation, where ATP synthase, which is just a special enzyme, uses energy from a proton gradient to phosphorylate ADP into ATP. So conceptually, substrate level phosphorylation looks like this. We have an enzyme. It's bound to another molecule called PEP. Now this enzyme transfers a phosphate group from PEP to ADP, and as you can see, thus we produce ATP and a molecule called pyruvate. So this is what substrate level phosphorylation physically looks like. So metabolism inside your cells is a multi-step process. In fact, there are 4 distinct processes that we're going to talk about today. And so to completely oxidize glucose, to get the most energy out of glucose, we have to go through glycolysis, the oxidation of pyruvate, the citric acid cycle, or it's also referred to as the tricarboxylic acid cycle, or the Krebs cycle, and finally the electron transport chain and what we call chemiosmosis. So let's just give you a brief overview. Glycolysis, in terms of where it's happening inside your cells, starts in the cytoplasm. Once glycolysis ends, you transfer into the mitochondria, where you have pyruvate oxidation. That is then followed by citric acid cycle, which is then followed by the electron transport chain and chemiosmosis. So, starts out in the cytoplasm and then again moves into our mitochondria. So first step is our friend glycolysis. Now, glycolysis starts by converting 1 molecule of glucose to 2-pyruvate. It is a 10-step biochemical pathway that occurs in the cytoplasm and has a net production of 2 ATP molecules by substrate level phosphorylation. In addition, it produces 2 NADH molecules by reducing NAD. So let's broadly look over what this, I should have made this bigger, I'm sorry. Give me one sec. me just make this a tad bigger so I can better show this off. Oh, shoot. All right. Hopefully that's a little bit bigger so it's easier to see. Go ahead. All right, so glycolysis again starts in the cytoplasm. You take that 6-carbon sugar, you do consume 2 ATP, creating, so you consume some energy during glycolysis. Now, there is, a part where you break apart the glucose, but at the end of the day, you produce 4 ATP. So it takes 2 ATP, but you make 4 ATP, thus your net is 2 ATP. So consumes, but produces 4, thus 4 minus 2 gives you 2. Now, as you'll notice along the way, we produce NADH, we also produce a second NADH, and then we also produce our friend pyruvate. Now, we can physically look at what this looks like from a 10-step process. And there are 10 enzymes associated with glycolysis. We're not going to walk through the steps. This is here for you to see what this looks like, to see how the chemical reaction is changing glucose into its final form of pyruvate. But again, the big thing to remember about glycolysis, glucose goes in, 2 molecules of pyruvate go out. Along the way, you reduce NAD twice into NADH, and you produce 2 ATP by substrate level phosphorylation. That's the big thing to remember. If you remember nothing else about glycolysis, just remember what goes in, what comes out, and what is produced. That's all that matters. I don't care that, step 1 is catalyzed by an enzyme called hexokinase, or the byproduct of step 8 is phosphoenolpyruvate. I don't care. I only care that you remember the checks and balances essentially here, right? How much comes in, what comes out, what do you produce, what are the important things? Now, for glycolysis to physically continue, right, NADH itself has to be recycled. And what does that mean? Well, it just means NADH needs to be oxidized. You need to strip the electrons from NADH, making it NAD again. Now, after glycolysis, you can essentially recycle NADH with either aerobic respiration, where we use oxygen as the final electron acceptor, or via fermentation. And this again happens when oxygen is not readily available. Now, so that's recycling NADH, but you'll see how we recycle NADH very shortly. Now, the fate of pyruvate is an important thing. Every time we go through glycolysis, we produce 2 pyruvate. And what happens to pyruvate depends on what we have in terms of oxygen. Now, when oxygen is present, pyruvate is oxidized to acetyl coenzyme A, or acetyl-CoA, which then enters the citric acid cycle. This is what we call aerobic respiration, and you'll see why in a second. Without oxygen, pyruvate is further reduced, typically to lactic acid or butane or ethanol or something like that, via the process of fermentation. But basically, it's just taking pyruvate to generate a little bit of NAD for glycolysis itself. And so let's just conceptually look at this. We have pyruvate with oxygen. One of 2 things happens. With oxygen, sorry about that, I was going the wrong way. With oxygen, NADH can be recycled with oxygen, so you reduce oxygen, thus oxidizing NADH, thus regenerating NAD. But the byproduct of oxidizing pyruvate is the production of our friend acetyl-CoA, which we will then send inside the citric acid cycle. Now, without oxygen, fermentation occurs. So fermentation can produce lactic acid. So if you like milk and, yogurts and kombucha, stuff like that, that's lactic acid fermentation. If you like wine and beer, that's ethanolic fermentation. So NADH has to be regenerated with oxygen. It's via respiration. Without oxygen presence, it's via fermentation. Now, in the presence of oxygen, pyruvate is further oxidized. Now, in eukaryotes, as we mentioned, it occurs in the mitochondria. For our prokaryotes, it does occur in the plasma membrane. So, prokaryotes don't have organelles, so it has to happen all inside the cell. Now, for every pyruvate molecule we stick into the pyruvate oxidation pipeline, we produce one CO2 via decarboxylation. I'm sorry, we produce one CO2 by decarboxylation by the enzyme pyruvate dehydrogenase. We produce one NADH, and we produce one acetyl-CoA. And now acetyl-CoA, if I'm not mistaken, I have a picture of it right here. This is what acetyl-CoA looks like. So this is what we start with. We produce, by converting pyruvate into acetyl-CoA, we produce CO2, NADH, and acetyl-CoA is the final byproduct. So that's what we're doing. But again, this happens inside the inner mitochondrial matrix here, inside our friend the mitochondria. So once we oxidize pyruvate to acetyl-CoA, we take acetyl-CoA and we oxidize it even further inside the citric acid cycle. Now, the citric acid cycle has many names. It could be called the TCA cycle or the tricarboxylic acid cycle. It could be called the citric acid cycle, as you see here. It also has been called the Krebs cycle after the gentleman who discovered it. Now, it's a biochemical pathway of 9 steps that are divided into 3 distinct segments. The first is where you can convert acetyl-CoA and oxaloacetate into citrate, i.e. that's why they call it the citric acid cycle. Then you have what we call citric rearrangement and decarboxylation. And then finally, the last step is to regenerate of oxaloacetate. Man, hard words. So let's just physically look at this. We start at the top here, because that's how cycles typically start. They start at midnight. Acetyl-CoA is fed in. There's a bunch of chemical reactions, but you'll notice a couple steps along the way. We produce NADH. We produce CO2. NADH, we produce CO2. We produce one ATP. We produce more NAD over here, but we also produce another electron-carrying molecule called FADH2. So the point of the citric acid cycle is simply to generate more NADH, again, more electron carriers, and we produce a tiny, tiny amount of energy, but we also produce carbon dioxide. Now, if you ever wondered why you breathe out carbon dioxide, it's because of the TCA cycle, the citric acid cycle. So as I mentioned, every acetyl-CoA molecule we stick inside the citric acid cycle generates us 2 molecules of CO2, 3 NADHs, 1 FADH2, 1 ATP, and it completely regenerates oxaloacetate. Because again, we start off with the first reaction itself is the reaction of oxaloacetate with acetyl-CoA. And again, I'm mostly concerned with the big picture here. This is the chemical reactions of what they physically look like over the course of this, right? We have the regeneration of oxaloacetate here, ATP generation down here, production of CO2 and NADH here. So you can see physically what's going on. But again, what really matters is the net products, the electron carriers, CO2, a little bit of ATP. That's pretty much it. But again, this is taking directly the products of glycolysis, oxidizing them to acetyl-CoA, and then using acetyl-CoA to generate more electron carriers and to generate some CO2 as well. The CO2 is not like they want to generate, it's just a byproduct of the chemistry. It's not like, oh, CO2 is great, but it's just a byproduct of chemistry. So after the citric acid cycle, so our 3-step process, glycolysis, pyruvate oxidation, and the citric acid cycle, every molecule of glucose that we stuffed into this 3-part process thus far, we've produced 6 CO2, 4 ATP, and then we've also generated 12 electron carriers. Now, the CO2, does one of 2 things. It diffuses out of your cell and into the environment if you're like a bacteria. Or if you're a eukaryote, it diffuses into your cytoplasm and then gets exported into your bloodstream. And then when it's in your bloodstream, it gets to your lungs and you breathe it out, right? That's the gist of it. There's more complex stuff behind it than that, but that's the general flow. So that stuff, not so important. The important things of what we've generated thus far is these electron carriers. Because remember, we're taking the energy stored in glucose and using electrons to transfer energy in a stepwise process. Now we carry that energy in those electrons in NADH and FADH2. The energy that's stored in here, about 53 kilocalories per mole, will then be used to generate ATP. So there's energy in here, but we're going to use that energy and make it into a biologically accessible form. And the way we do this is by one of the most ingenious biological systems ever created, in my opinion, of course, which is the electron transport system, or the ETC for short. Now, the ETC is a series of membrane-bound electron carriers, proteins and the like, which is embedded in the inner mitochondrial matrix in eukaryotes. In the prokaryotes, it's on their cellular membrane in what's called the periplasmic space. But we're focusing on eukaryotes here. But just know that the process is essentially identical between prokaryotes and eukaryotes. Now, the electron transport physically takes the electrons from NADH and FADH2, transfers them to protein complexes in the ETC, and each one operates a proton pump, driving protons into the intermembrane space of the mitochondria, creating a proton gradient. And it allows, this process allows electrons to move from protein to protein within this complex. So let's look conceptually what I mean by this, just like big picture stuff. Again, we're happening inside of mitochondria here. We have the inner mitochondrial membrane here. We have a couple enzymes. Now, you'll notice that we have NADH. We combine that with a hydrogen ion. It transfers 2 electrons to our very first enzyme, which we call NADH dehydrogenase. It's called that because that's what it does. And what it does from there is it transfers those 2 electrons to what we call a quinone, which is denoted as Q. It's just an intermediate electron carrier. You'll notice that it pumps hydrogen ions from the mitochondrial matrix into the intermembrane space. Then we have electrons transferred from FADH to that quinone as well, producing FADA. So this is getting oxidized. Then the quinone transfers things to the next complex, which transfers things to the next complex and to the next complex. And what we're talking about transferring here is electrons, right? We're moving electrons. And you'll notice that along the way, we pump protons across the gradient. And then you'll notice that we finally get to oxygen, which is our terminal electron acceptor, and it is the final step in this process. We transfer electrons from complex to complex, from protein to protein, and eventually we transfer those electrons to our friend oxygen. But where did we get those electrons from? We got them from NADH. Where did NADH get those electrons from originally? From glycolysis, from pyruvate oxidation paired with the TCA cycle, right? So why does that matter? Why do we need to transport electrons? Well, it's really to drive the final process, which we call chemiosmosis. Throughout this step, we're pumping protons across from the mitochondrial matrix into the intermembrane space. But why? Well, we create a gradient. And a gradient, as you might sort of think about conceptually, is really like water behind a dam. If you have water behind a dam, well, that water has the potential to flow. The same way with protons. If you have a gradient, they have the potential to flow. And the same way, if you have a hydroelectric dam, if water flows through a hydroelectric dam, it generates energy. Now, in our case, if protons flow through a special type of protein, they have the potential to generate energy. So creating a proton gradient via the electron transport chain creates a gradient of potential energy by which we can use to generate kinetic energy. And that kinetic energy that we generate has the potential to generate ATP. And that process is what we term chemiosmosis. Now, the enzyme involved here is aptly named ATP synthase. It is the turbine in the hydroelectric dam. It is what takes that gradient of protons, basically spins, and generates energy from that. And that energy is then used to produce ATP. So let's dive into this a bit more. Now, during the electron transport chain, we accumulate protons in the intermembrane space. Um, and remember that membrane themselves, they're basically impervious to ions. So once you create a proton gradient, it stays there until you release it manually. Now, protons themselves can only re-enter the matrix space through ATP synthase. ATP synthase, ATP, synth meaning generating, so ACE meaning enzyme, so it's an enzyme that generates ATP. How do you generate ATP? You phosphorylate ADP. There's a typo here. It should say ADP and phosphate. Sorry. Now, by using ATP synthase to generate ATP from ADP in a phosphate group, we call this chemiosmosis. So let's just bring this back to big picture before we talk a little bit more about ATP synthase. We started with one molecule of glucose. Glucose was oxidized fully to pyruvate in glycolysis. In the process, we produced ATP and NADH. We took the resulting pyruvate, stuffed it into pyruvate oxidation. We produced carbon dioxide, we produced more NADH, and we produced acetyl-CoA. We took the acetyl-CoA, we stuffed it inside the citric acid cycle. Inside the citric acid cycle, we produced more ATP, we produced CO2, we produced more NADH, and we produced FADH2. Now remember, FADH2 and NADH are just electron carriers, but we carry electrons because electrons have energy. These molecules have potential energy. After the citric acid cycle, these high energy electron carriers transfer their electrons into the electron transport chain, where they are used to pump protons across the gradient into the intermembrane space here. So from the matrix into the intermembrane space. And that gradient allows ATP synthase, which is, again, think of it like a hydroelectric dam turbine, to generate 28 ATP per go-round, essentially. So that's the flow of information. If you don't remember anything from this lecture, at least from this first part, remember this figure. This is the big picture stuff that you want to remember. What is going on? Big picture. Now, we've skipped over a lot of stuff, right? We didn't look at the enzymes. We didn't look at how all this is controlled. There's a lot of stuff we didn't cover. Because I want you to focus on big picture things here. What is an electron carrier? What is the redox reaction? Why does it generate so much energy? So let's dive into this enzyme, ATP synthase. It's a cool, I have a picture, good. It's a cool enzyme. It really is. Now, ATP synthase is a tiny rotary protein. And I mean that. It's like a little rotor. It has two subunits, basically. It has the F0 complex, which is a membrane-bound complex, and it has the F1 complex, which we call the stalk and knob, which has enzymatic activity. Protons themselves physically flow through the F0 channel, which causes F0 to rotate. Again, it's a little rotary. And this mechanical energy of rotation changes the conformation of F1, which we call the catalytic domain. Now, what does changing the conformation mean? Remember, it's changing the shape. So let's conceptualize this. We have that F0 or the F0 here. It's membrane bound. Then we have the catalytic head. They're bound together by a protein, which is called the F clamp. Well, we're not going to talk about it, but just figured I'd mention of what this sucker is. But again, through the whole electron transport chain, we pumped a bunch of protons from the matrix into the membrane space. Now these protons are going to flow through ATP synthase. This causes ATP synthase every time a proton flows through to essentially rotate. Just think about like spinning, like if you ever use like one of those little fidget spinners, or like you like spinning the wheels on your bike. That's the same idea. It causes this protein to literally rotate. It causes it to rotate, which again causes a conformational change in the catalytic head. And that conformational change allows the catalytic head, it gives the catalytic head energy that you get by rotating something to phosphorylate ADP into ATP, thus taking mechanical energy from motion into chemical energy, which is ATP. And remember, ATP is just the energy currency of our cell. So the past 20 minutes, we've basically been building to this one moment where we turn a rotary protein and it takes mechanical energy and turns it into chemical energy. And if that's not like the coolest end of a pathway that you've ever learned about, I don't know. Right? I mean, that's cool, right? It's like a biological hydroelectric dam, essentially. But so that's the end. Now, the energy yield of respiration is about 32 ATP per glucose molecule for bacteria and about 30 ATP for eukaryotes. We are simply less efficient. I think humans get about 24 on average per molecule of glucose. Actually, I think if you're obese, you get less than that. I think you get like 15 per glucose molecule if you're an obese human. I think that's what it is. Don't quote me on that. Now, we can look at what we call the PO ratio, not the BO ratio, but the PO ratio, which is the phosphate to oxygen ratio, which is simply the amount of ATP synthesized per oxygen molecule. Remember, we needed final electron acceptor was oxygen. So we can look at what we call theoretical and direct calculations of PO, and it's been contentious and has actually changed over time. So it's kind of hard, but we can look at the theoretical yields. You know, we produce 2 ATP, and then, you know, ATP in chemiosmosis and the citric acid cycle and all that fun stuff. So the total yield is potentially 32. But you don't always get that, right? As I mentioned, if you're obese, sometimes if you have like conditions in your mitochondria, you don't get that 32. So the ratio of phosphates, ATP produced to oxygen is variable. But the maximum theoretical is 32 and 30. Now, respiration is controlled. Like everything inside you, you just don't respire 24/7. Right? Your cell, every cell in your body isn't continuously respiring. Now, do you need energy when you sleep? Of course you do. But how much energy you need is dependent on the activity. So your cells have to regulate how much respiration occurs. Now, the regulation of respiration is feedback inhibition. Now, there are 2 key points by which we control respiration. The first is in glycolysis. An enzyme called phosphofructokinase is actually allosterically inhibited by ATP or citrate. So this enzyme is one of the early steps in glycolysis. I think it's step 3, if I'm not mistaken. It can get bound out by excesses of ATP and citrate to basically shut it down. And if you shut down this enzyme, you shut down glycolysis. Now, you can shut down pyruvate oxidation and the citric acid cycle by targeting the enzyme pyruvate dehydrogenase. And pyruvate dehydrogenase is inhibited by very high levels of NADH. You can also inhibit the enzyme, one of the early ones, citrate synthase, by high levels of ATP as well. So we can use the byproducts of glycolysis and citric acid cycle and pyruvate oxidation to limit how much we physically do. Because if I have a lot of ATP, I don't need to keep making ATP, right? ATP only lasts for a little bit, right? So we have to regulate it. And nice pathway, you know, sort of molecule that shows like, okay, here's where I produce ATP, here's where I produce these things, and here is where these enzymes are inhibited. But again, it's all about allosteric inhibition. They bind to the not the active site, but the allosteric site on these enzymes, causing them to be stopped, thus stopping the process. So really handy, you know, conceptual model of everything I just said on this slide. All right. So what we have discussed is metabolism that's in the presence of oxygen, aerobic metabolism. But anaerobic metabolism and fermentation are out there. So anaerobic metabolism, as I've mentioned, for the terminal electron acceptor, they use inorganic molecules like nitrogen and sulfur in place of oxygen in their electron transport chains. Fermentation uses organic molecules as the final electron acceptor. So just want to make mention of a few of these anaerobic respirations because, like I said, I did all this work when I was a graduate student. So one of the cooler ones, which is only done by archaea, is methanogen or methanogenesis. They reduce CO2 to methane. And so methane, as you probably know, is a greenhouse gas. It's also found in cow burps, not in cow farts. And so cow burps produce methane because of the archaea inside their stomach. But again, they're reducing CO2 to methane. Methane is, one of the byproducts. We have sulfate reducing microbes. So they take inorganic sulfate as the terminal electron acceptor, reducing it to hydrogen sulfide. Now, if you if you've ever been to the ocean and it smelled kind of like rotting eggs, or it had really bad smell at low tide, you can thank sulfate reducing bacteria. They produce hydrogen sulfide, which is the smell of rotting eggs, which is that nasty smell at low tide, or that nasty smell in the ocean that the ocean typically gets. And you can just to sort of see this, sulfur respiring organisms are everywhere. They're pretty cool. You can find them inside of hot springs like this. You can physically see them. Like all this yellow stuff is sulfur respiring organisms. So just as a note. Now, arguably the more important anaerobic metabolism is fermentation. Now, fermentation, as I've mentioned, reduces organic molecules to regenerate NAD. Now, there are two broad classification. Well, there's many classifications of fermentation, but the ones we're going to discuss are ethanolic fermentation, where CO2 and ethanol and NAD are physically produced. This occurs in yeasts. I'm pretty sure it's just yeast. I don't think any bacteria do it, but pretty sure it's just yeast. And then lactic acid fermentation. Now, this occurs in your cells. So you do lactic acid fermentation, but it's also done by a group of bacteria called the lactic acid bacteria. So if you like yogurt, if you like kimchi, if you like kombucha, you rely on lactic acid fermentation to make it taste good. But again, it does occur in your cells. And remember, in this case, the electrons are transferred to NADH, from NADH to pyruvate, which eventually. So let's just look at these basic pathways to see what alcoholic and lactic acid fermentation looks like. Glycolysis occurs, we produce 2-pyruvate. That 2-pyruvate is then used to produce CO2 and acetaldehyde, and then acetaldehyde is then used to produce ethanol. And we basically, remember, the whole point of fermentation is to regenerate NAD for glycolysis. So they ferment it to ethanol, oxidizing NADH back to NAD. Thus regenerate NAD. Now, lactic acid fermentation, glycolysis occurs, we produce pyruvate. Pyruvate is directly oxidized to lactate or lactic acid, and that lactic acid, remember, is produced simply to regenerate NAD by oxidizing NADH. Whew. So we've thus been talking directly about the breaking down of sugars, right? Everything started with glucose, but you guys know that you can eat other things, right? You can fats, you can eat sugars, and you can get energy from those things. Now, amino acids themselves can undergo a process which we call deamination to remove their amino group. And then the remainder of the amino acid is then converted into a molecule that is capable of entering glycolysis or the citric acid cycle. So just as a note, we have alanine. One of our amino acids, alanine, is typically converted to pyruvate, or we can take aspartate and convert it to oxaloacetate, which you should remember is part of the TCA cycle. So let's just look at what deamination looks like. So we have glutamate, so one of our amino acids. We have that standard structure. We have the backbone here, and this is the side chain. Now, deamination involves the removal of the amino group from the backbone itself, and that amino group is then used to make urea. And by removing the amino group, the NH3, we produce alpha-ketoglutarate. Now, if you ever wondered why you have urea in your pee, one of the reasons you have urea in your pee is because of deamination. Breaking down proteins, i.e. breaking down waste, produces urea pretty commonly. Now, this alpha-ketoglutarate is then used in a part of your, your cellular respiration cycles. It can go directly into part of the TCA cycle, right? It's the first step in the TCA cycle. Now, fats can also be catabolized. So fats are physically broken down into fatty acids and glycerol. Now, the fatty acids themselves are converted to acetyl groups by what we call beta oxidation. This is an oxygen-dependent process. And the interesting thing is, as you respire a 6-carbon fatty acid, it actually yields 20% more energy than a 6-carbon glucose. So let me show you what beta oxidation looks like. So we have a fatty acid here. We use CoA to convert it into a fatty acid CoA. We generate FAD along the way, and then we generate NADH along the way, eventually producing a fatty acid CoA. And that fatty acid CoA, part of it then can be used to make acetyl CoA. You'll notice that this is what acetyl CoA simply is. And that acetyl CoA can physically then be stuck into the citric acid cycle, thus to generate a little bit of ATP, as well as generate electron gradients, right? Now, thinking about this, right, we only consume 1 ATP, right? Whereas to do this to glucose, we have to consume 2 ATP, right? So we do consume less glucose, which is how we get slightly more energy. Now, this is what energy extraction pathways, we're not going to walk through it, but I just want to sort of give you the idea of how you can break down nucleic acids, proteins, sugars, and lipids and fats. What are the processes that are used to physically stick these things that you eat into your metabolism? So nice handy figure here for you to digest at your wheel, because we've already covered a lot today, so I'm not going to poke more at that. And there is some information here that you can digest on your own about the evolution of metabolism as a whole. So sort of this hypothetical timeline. All right, so that's respiration. Now, the counterpart to respiration is our friend photosynthesis. As I mentioned before, the vast majority of energy for life on the planet gets energy from the sun and from photosynthesis. Now, you'll notice that the reaction for photosynthesis, CO2 plus water plus light, generates sugar, so glucose, oxygen, and water. Now, you'll notice that this is the exact opposite of of respiration, right? Respiration takes this and this and this and makes CO2 and energy. So respiration and photosynthesis are basically two sides of the same coin. They're just basically just opposites, right? Which is kind of nice in terms of remembering what's going on. Now, oxygenic photosynthesis, i.e. photosynthesis in the presence of oxygen, there is an oxygenic photosynthesis, so photosynthesis carried out in the absence of oxygen. But photosynthesis in the presence of oxygen is carried out by cyanobacteria, algae, and all land plants inside their chloroplasts. So photosynthesis has a couple different stages. We break them down into 2 key parts. We call them the light-dependent reactions or the light-independent reactions. So the light-dependent reactions, as their name tells you, do physically require light. They capture energy from the sunlight, they make ATP and reduce NADP to NADPH. You'll recognize NADPH as being very similar to NADH, and you'd be absolutely right. They serve the same function. And the second step, which we call the light-independent reactions, or also carbon fixation, doesn't require light and uses the NADPH and ATP to synthesize organic molecules from CO2. This should be a lower subscript 2, not a superscript, just as a note. So that's the sort of the major stages. Now let's talk about the chloroplasts. We've already discussed these a bit, but we'll just give a refresher. Now the chloroplasts have their outer membrane, and they are a 2-membrane system. So they have the outer membrane and the inner membrane. And you'll remember that they have the these stacks called thycaloids. And these thycaloids are covered by the thycaloid membrane, as you would imagine. Now, the thycaloid membrane contains chlorophyll and other photosynthetic pigments, and all these pigments are clustered into photosystems. Pretty straightforward, right? And there's other parts of the anatomy here we don't care about. We really just care about this chlorophyll and these photosystems. That's really all we care about in terms of this. So let's give ourselves a broad overview of photosynthesis. We have those photosystems which are embedded in the thycaloid membrane that capture sunlight. and they capture that sunlight again to do all that, all that fun stuff, right? They take CO2. Um, I'm sorry, they produce oxygen, they produce ATP, they produce NADPH, which is then transferred to the light-independent reactions, where we fix that carbon, producing organic molecules. So that's the flow of information. Energy from the sun is captured. It's then used to generate ATP and NADPH, and those two in turn are used to fix carbon dioxide into organic molecules, i.e. glucose. All right, so historically speaking, John Baptiste de von Helmont in the 1580s to the early 1600s demonstrated that the substance of a plant was not produced from the soil. So, plants take mass from the air, right? Air has mass, right? So they take their mass from the air, right? They don't take mass from the soil. Um, uh, Joseph Priestley in the 1700s showed that living vegetation adds something to air, and then Jan Eigenhaus in the 1700s proposed that plants carry out a process that uses sunlight to split carbon into oxygen and carbon as well. So sugar and oxygen. And so that's the sort of the background of it. Now, one of the more important guys along the way was F.F. Blackman in the 1800s and early 1900s. He came to the conclusion that photosynthesis was a multi-stage process, only of which one portion uses light directly. So he's the guy that came up with the light versus the dark reactions, i.e. light dependent and light independent. He also proposed that enzymes were involved. Fast forward to the late 1800s and into the 1900s, C.B. von Neal found that purple sulfur bacteria, which are bacteria that do anoxygenic or photosynthesis in the absence of oxygen, were a thing. So their chemical reaction looks a little bit like this. So similar to photosynthesis, but distinct. Robin Hill also demonstrated very similarly processes as von Neal. So that's a little bit of history. I think it's nice to have some perspective. Photosynthesis is, we're talking about it in this course like it's settled science, but we learn so much about photosynthesis all the freaking time. It's crazy how much we still don't know about photosynthesis, but again, it's super important. Now, photosynthesis is reliant on pigments, and these are molecules that absorb light energy within the visible range. Now, the light that comes from the sun is a form of energy, and the energy comes, in terms of light, comes as what we call a photon, and photons act as a discrete bundle of energy, so they have kinetic energy, and we use what's called the photoelectric effect, which is the removal of an electron by a molecule, from a molecule by light. So we use the energy of light to remove an electron, which thus up an electron, which is just energy. So we take energy and light to free up an electron to provide the photoelectric effect. And as you guys should probably get the idea from now, the production of free electrons is kind of important, right, for metabolism. So this leads us to the electromagnetic spectrum. Now, the energy released from the sun has varying wavelengths. So I'm sure you probably have heard of this before, but light travels as a wave. And so the longer the wavelength, the less ionizing it is. The shorter the wavelength, the more ionizing it is. So wavelengths greater than the visible spectrum, i.e. what we can see, red, orange, yellow, green, indigo, violet, those are ionizing. They are dangerous. So X-rays and UV and gamma rays are very dangerous forms of energy. They are emitted by the sun. Energy like infrared and radio waves, they're safe. They don't harm you at all. So just make mention of this. 5G radiation, 4G radiation falls in this category here. It's safe. It's non-ionizing. Just as a note for all those people that say otherwise. But again, all the energy that you from the sun that you physically can see falls into the visible spectrum, i.e. 400 nanometers to 740 nanometers. And this is where photosynthesis gets its energy from. It takes wavelengths of light in this wavelength and uses it to derive energy. So this leads us to what we call the absorption spectrum. When a photon strikes a molecule, the energy itself is either lost as heat or absorbed by the electrons in the molecule. Now, when something absorbs energy from light, it causes the electrons to increase their energy level. And that makes intuitive sense, right? If you add energy to something, it gets energy, right? And that's all sunlight is, right? It's just energy. So if we add energy to a molecule from the sun, it just causes that molecule to gain more energy. Now, the absorption spectrum is the range and efficiency of photons a molecule is physically capable of absorbing. And why am I saying this? Well, because photosynthesis relies on absorption of light. And it relies on the absorption of light by specific molecules, which we call chlorophyll. I'm sure you guys have heard of chlorophyll at some point. Photosynthesis also relies on the absorption of light by what we call carotenoids and other types of photosynthetic pigments to help increase photosynthetic efficiency as well. But what you'll notice is that we have low light absorption and high light absorption, and then we have the visible wavelengths here on the bottom. Chlorophyll B, which is in the dotted line, and chlorophyll A, which is in the solid dark line, and then we have carotenoids, which are in the gray line here. You'll notice is that they have absorption at about 420 nanometers, and then again at 670 nanometers in that ballpark. And then chlorophyll B has a high peak at around 450, and another one at about 630 or so. So they absorb light at very specific wavelengths, not just any old wavelength in the spectrum, but very specific wavelengths. Now, photosynthesis, as I mentioned, relies directly on the pigment chlorophyll. There are many, many types of chlorophyll out there that photosynthesis. Remember, chlorophyll is the pigment by which organisms rely on to absorb energy from the sun, i.e. to capture the energy in the sun. So chlorophyll A is the main pigment we see in bacteria and plants, and it's the only pigment that can physically directly convert light energy to chemical energy. So chlorophyll A is where it's at in terms of capturing energy from the sun. And as I mentioned before, it absorbs violet, blue, and red light. Chlorophyll A also reflects green light, which is why chlorophyll looks green, which is why plant leaves look green. Anything that a substance reflects back and doesn't absorb is the color of that substance. So if I have a blue phone or a blue piece of paper, that blue piece of paper is reflecting blue light back to your eyes. It's absorbing all the other wavelengths, it's reflecting blue light back to your eyes. Now, chlorophyll B is what we call an accessory pigment or a secondary pigment. It absorbs light wavelengths that chlorophyll A does not absorb, but again, not as important into this whole process of this. Now, chlorophyll has a pretty basic structure. It has what we call a porphyrin ring. Now, this is a pretty complex ring structure with alternating double and single bonds with a magnesium ion stuck direct dab center in the middle. Now, what happens here is photons of light excite electrons in the ring, and the electrons are then shuttled away from the ring. So let me show you what our friend chlorophyll looks like. We have the porphyrin ring, we have this tail of hydrocarbons, and magnesium is directly at the center. Now, chlorophyll A and chlorophyll B, very, very similar. This little R part just differs slightly. So chlorophyll A has a CH3 and chlorophyll B has a CHO. But basically, electrons hit this ring and are shuffled down into the hydrocarbon tail. That's how things really work. I'm sorry, the wavelengths of light excite the electron shuffled away from the porphyrin head. I'm actually going to skip this. Now, in addition to chlorophyll, I just want to make mention of carotenoids and other accessory pigments, including phycoxyloproteins. Now, these are accessory pigments which basically help scavenge free radicals, so they help protect the photosynthetic apparatus. They also absorb other wavelengths of light that are not ideal for photosynthesis. And so the bottom line for these types of pigments is they just help improve photosynthetic efficiency. They're present inside the thylakoid membranes, but they're not directly involved with the production of the stuff we're going to talk about. But again, they are kind of important. And I wish I had given this lecture like a month ago or so, these carotenoids, these accessory pigments, give leaves their awesome colors in New England and other places. So plants, as I mentioned, are green because of chlorophyll, but when plants pull their chlorophyll in during the colder months, you're only left with the accessory pigments. They're pulled in after the fact. So when a plant goes from, like, say, green to red, you're just left with accessory pigments, which are red. Or maybe you're left with yellow ones, which are yellow colored. Or maybe you're left with orange ones, which accessory pigments are orange colored. But eventually the plants pull all of their pigments in, leaving the plant to be brown, right? The plant leaves to be brown. So in case you wondered how fall works here in New England, it's because of accessory pigments. Now, with all that stuff, light has to physically be captured, right? That's the first step in photosynthesis. Now, light is captured by our photosystems, which are comprised of two distinct components. The antenna complex, which contains hundreds of accessory pigment molecules, as well as chlorophyll. And they gather photons and they feed that captured light energy to the reactant center, which has one or more chlorophyll A molecules, which then in turn passes the excited electrons out of the photosystem. Now, the antenna complex is also called the light harvesting complex. As I mentioned, it captures photons of light and channels them into the reaction center for the chlorophyll. Now, in the chloroplast, the light harvesting complex consists of a web of chlorophyll molecules linked together and tightly held in the thylakoid membrane by matrix proteins. So let's look at this photosystem structure. We have the chlorophyll molecules, we have the thylakoid membrane, and this is our photosystem. Photons come in, chlorophyll absorbs it. Now, what happens to the light energy? It's transferred from to chlorophyll to chlorophyll to chlorophyll, eventually reaching reaction center. Once it reaches the reaction center, and when I say reaches, I mean the energy, right? The energy from the sun. The energy from the sun reaches the reaction center. It takes the energy and transfers that energy to an electron donor. That electron donor, it takes the energy to pull an electron from the electron donor, creating an electron carrier, i.e. the electron acceptor. So this would be like NADH, right? So that's the sort of thing. Light comes in, the energy bounces around until it gets to the reaction center, which is then used to stick an electron on something else to transfer the energy in the form of an electron, very much like respiration, to something else. So let's look at that. Well, let me talk about this first. Now, the reaction center, remember, it's it is a transmembrane protein pigment complex. And so when a chlorophyll in the reaction center does absorb a photon of light, an electron is then excited to a higher energy level, i.e. it gains energy. Now, this light energized electron, which has more energy, is then transferred to the primary electron acceptor, thus reducing it. Now, oxidized chlorophyll then fills its electron hole by oxidizing a donor molecule. So what does that look like? Well, we have this energy conversion. Light is absorbed by our system. That then is used to excite an electron to a higher energy state, and then transfer that to basically take that electron, transfer it to an electron acceptor. Then our chlorophyll, as you'll notice, is lacking an electron, is thus positively charged. It then scavenges an electron from an electron donor, thus bringing it back to its original system, where it can then absorb light, transfer its electron that's high energy, and rinse and repeat the process over and over and over again. All right. So this leads us to our light-dependent reactions, where we have the capture of light, which is the primary photo event, which we've just discussed pretty much in depth. This leads us to the charge separation, where the energy is transferred to the reaction center. The electron is excited, transferring an excited electron to an acceptor molecule. So this is what we just talked about, 1 and 2. Then we have the electron transport chain, very similar to the electron transport chain we just talked about, but we basically move electrons to carriers to reduce NADPH. And then we have chemiosmosis to generate ATP. Now, I'm actually going to skip this. We're going to not talk about that. Now, chloroplasts have two connected photosystems. They have to carry out oxygenic photosynthesis. Now, they have photosystem 1, or P700, and they have photosystem 2, which is P680. Now, these work together to carry out what we call a non-cyclic transfer of electrons that is used to generate both ATP and NADPH. And there is evidence, but we're going to skip over that. Now, as I mentioned, our two photosystems work together. Now, photosystem 1 transfers electrons to NADPH, reducing it to NADPH. The electrons lost from photosystem 1 are then replaced by electrons from photosystem 2. Photosystem 2 oxidizes water to replace the electrons transferred to photosystem 1. And the two photosystems are connected by what we call a cytochrome B6F complex. And let's just look at what this looks like. So we have photosystem 1 and photosystem 2. They're named by which they were discovered. So this was discovered first, then this was discovered. They're not named by the order by which they operate. But it does, again, start with mostly, it starts with photosystem 2 and then goes into photosystem 1. So they're named 1 and 2 not because of the order, but because of how they were discovered. So starts with photosystem 2 in the reaction center. Energy in the form of light comes in, goes to reaction center, excites an electron, causing it to jump up an energy level to be transferred to the electrons, an excited electron carrier, which then passes the electrons through a couple of different things. So the first is what we call a plastoquinone. It's just a protein. And then eventually translated to the B6F complex, where it generates a proton gradient. So that should sound familiar. And then that B6F complex transfers the electron again to plastocyanin, and then finally making it to the reaction center of photosystem 1. Photosystem 1 then absorbs more light, exciting the electron, passing that electron to the reaction center, passing the electron to ferredoxin, passing it to an enzyme called NADPH reductase, which creates NADPH from NADP. And that's our process of photosynthesis. Nothing, well, I don't know, kind of crazy. It's more complex than respiration, certainly, but nothing in terms of new concepts, new words, but definitely not new in terms of relative to respiration, of course. As I've mentioned, the BF complex is a proton pump. It's embedded in the thylakoid membrane. It does generate a proton gradient for us. And in terms of why we generate, oh shoot. No, I'm not trying to sign in. Sorry about that. Someone trying to sign into my, anyways, no, not important. So in terms of what this looks like in 3D space, right? We're generating a proton gradient, right? And remember that proton gradient is generated by that B6F complex right smack dab in the middle between photosystem 1 and 2. Now, in terms of what this conceptually does, remember we're just pumping protons, right, from the outer membrane into the inner membrane. Now, those protons are used exactly the same way as they are used in respiration, but they're used to generate ATP, right? They have ATP synthase. It's exactly the same as it is in animal cells and the mitochondria. Does the same job, proton gradient to generate ATP from ADP. But again, it's kind of this funky thing, right? Instead of like using like glycolysis and stuff like that, right? It's using the energy from light to generate the proton gradient. So kind of conceptually funky, but at the same time, it should be conceptually very familiar. Transferring electrons to generate a proton gradient, to send through ATP synthase to generate ATP. Now, the production of ATP, in terms of this process, now our sort of this process is generically called photophosphorylation. So we talked about oxidative phosphorylation. This is called photophosphorylation because we're using light to generate ATP, to phosphorylate something. Now, phosphorylation generates NADPH, which is our electron carrier, and ATP. And it uses all this stuff to build organic molecules. That's all we're doing. That's all we're really doing. And we're doing all this, we're generating all this ATP, NADPH to stick into carbon fixation, which has been named the Calvin cycle. Now, Calvin cycle is basically just used to build carbohydrates. They take the energy from chemiosmosis in the light-dependent reactions to basically pair it with the reduction potential of NADPH from photosystem 1 to build carbohydrates. So take carbon from the atmosphere and fix it with the energy derived from the first part of photosynthesis to make sugar. So the Calvin cycle was named after a gentleman by name of Melvin Calvin. He called it C3 photosynthesis. And the reason he called this is because the first intermediate molecule has 3 carbons. But the key step in this process is carried out by an enzyme called ribulose 1,5-bisphosphate, or Rubisco, or RUBP. When you do that in-class exercise next week on your own, you'll see Rubisco. But the key step in here is the attachment of CO2, a 5-carbon sugar, i.e. the carbon fixation part. So there are 3 phases of the Calvin cycle. We have carbon fixation, which RUBP plus CO2 to produce PGA. We have reduction, where PGA is reduced to G3P. And then we have the regeneration of RUBP, where G3P is used to regenerate RUBP. So 3 turns of the Calvin cycle incorporate enough carbon to produce a new G3P, and 6 turns incorporate enough carbon for 1 single glucose. So it takes 6 cycles of what we're about to discuss to produce a single molecule of glucose. So let's conceptually look at this. We have our 3 phases: carbon fixation, reduction, and regeneration. 6 molecules of carbon dioxide are imported. The enzyme Rubisco makes 3 PGA. Then we consume 12 ATP. We consume 12 NADPHs, producing glyceraldehyde 3-phosphate, or G3P. Remember, every turn of this produces G3P. Once you produce enough G3P, I want to give you the right numbers. So basically, we need to produce 6 G3P 2 G3P to produce 1 glucose molecule. So that takes 6 full turns to produce. Lots of turning, but again, I don't care so much about the specifics. There's a lot of specifics here. I mostly care what's consumed, what's produced, and those big name molecules. Now, remember, the output here isn't glucose. The output is G3P, right? Glyceraldehyde 3-phosphate. It's used to form sugars in plants. It's in the sugar, it's actually sucrose, and as we've mentioned before, sucrose is a disaccharide, meaning by linking fructose and glucose together. Sucrose itself, you link a bunch of sucroses together, it actually is used to make starch, and starch is a storage molecule for plants. All right, so this is all sort of blanket called our energy cycle. Now, photosynthesis uses the products of respiration as starting substrates. Respiration uses the products of photosynthesis as starting substrates. So they are linked together. Now, the production of glucose from G3P even uses part of an ancient glycolytic pathway, basically run in reverse. So everything is linked. I want to just sort of put that together. Now, photosynthesis and respiration in plants occurs within the same cell, right? And they are linked together in some way, shape, or form. So as a plant is doing its thing, it's producing oxygen, which it uses in the its own electron transport chain. It's also producing CO2, which it uses in the Calvin cycle. Once you produce glucose, you can then generate pyruvate for your citric acid cycle in your electron transport chain, and it's all linked together, kind of nice. So when we link these together, we call them photorespiration. Well, because we're just about out of time, I want to make sure I talk about the important stuff. Yeah, so I want to just talk briefly about this before I break off. So those are sort of the big things about photosynthesis. Now, we talked about C3 photosynthesis, production of carbon, of sugars by the Calvin cycle. Now, there are two other types of photosynthesis, which we call C4 and CAM. Now, C4 and CAM photosynthesis are kind of cool. They're generally only seen in like desert plants and places that have low water. So, corn, for instance, is a C4 plant. So I just want to make mention of that. Now, C3 and C4 photosynthesis and CAM photosynthesis, they're really similar, right? They take CO2, but the key difference between C3 and C4 is that they're separated in space and time. So in C3 plants, everything is happening inside of a plant cell. Now, in C4 plants, such as corn, or maybe something like a cactus, it is separated into two distinct compartments. In C4 plants, we have mesophyll cells, as you can see here, around sort of the circle here, and then we have bundle sheath cells here in the center. And so photosynthesis there is separated into two distinct cellular compartments. Whereas in C3 plants, all this happens inside this mesophyll cells right here, just as a note. Now, C4 plants, they fix carbon using an enzyme called PEP carboxylase. This happens again in those mesophyll cells. Now, this itself, from carbon dioxide, produces oxaloacetate, which is then converted into malate. And once it's converted into malate, is then transported into those bundle sheath cells. Now, once it makes it into those bundle sheath cells, the malate is then decarboxylated to produce pyruvate and CO2, and then they use that CO2 to produce sugars. So just as a note in terms of what this looks like, the differences between C3 and C4 plants, we have mesophyll cells, which again take CO2, convert it into malate, which malate then can be exported into bundle sheath cells where the Calvin cycle can then happen. And that's sort of the big difference. It is a little bit more energy intensive. It requires an extra 12 ATP, but it does give plants a pretty big advantage in dry and hot climates because the C4 pathway uses less water, thus, and it also relies on your plant cells being open for the absorption of gas for a shorter amount of time. So it is much more water conserving than the C3 plants are. So, and that's all I really wanted to make mention of. We'll skip the CAM plants because they're less important. I just wanted to make mention of the C4 plants and how they are different from C3 plants, which we spent the bulk of our photosynthesis time talking about. All right, that was a literal crap ton of stuff. So I'm going to stop recording.